Today's lesson is going to be about blood vessels. So we'll be talking about the arteries and the veins, differences between them. So what do arteries do? What do veins do? Because of their job, they're built differently. Blood vessels, arteries, arterioles, which are smaller, capillaries, venules, veins, form a closed circuit, closed system that carries blood away from the heart to the cells and then back to the heart. So arteries we'll start with first. Arteries are going to take blood away. Arteries away from the heart. Because they're taking blood away from the heart, they're taking it when the blood is being contracted with a large amount of force. So the force of blood in arteries is bigger than in veins. Therefore, they're stronger, they're more elastic, they actually keep their shape a little bit better. They're adapted for carrying that high pressure blood. Arteries become smaller and smaller as they branch off and divide and give rise to arterioles. So basically, your aorta comes off of your heart and it branches and branches and branches into smaller and smaller parts. So that aorta leads to or branches into most of the arteries that goes to your cells. So when we look at the construction of an artery, what it's built like, endothelium, the inside, is called tunica internal inside, tunica media, middle, and that's made up of smooth muscle. And then tunica externa or tunica external is made up of connective tissue. So there's three layers to your arteries. Endothelium, simple squamous tissue. It's basically a type of epithelium. Inside it creates a very smooth surface so that clotting does not happen. It secretes chemicals that prevent your platelets from grouping up and clotting. And it also secretes certain substances that will regulate how fast your blood flows. Tunica media is the thickest layer of the arteries, and that really kind of gives it its shape. Arteries will hold its shape much better, where a vein, if there's nothing in the vein will kind of become flat. So the tunica media is the thick layer that kind of helps it hold its shape. Tunica external attaches the artery to the surrounding tissues that are around it. Arteries can constrict and arteries can dilate. So they're capable of vaso, means vessel. So vasoconstriction, constriction of the vessel. So when my nervous or sympathetic system tells me that I need to constrict, that blood vessel will get smaller to slow down blood flow. When those impulses are inhibited, vasodilation, vessels dilate, then it's going to allow more blood to move through, and that opening and closing or constricting and dilating can regulate the blood flow and your blood pressure. As these arteries and arterioles get smaller and thinner, they approach the capillaries. So the smaller they get, the closer to the capillaries. Their vessels themselves get thinner because the pressure is not quite so high. So I like this picture because it shows the difference between them. If I look at the outside lining, here's an artery, there's the vein. The outside lining, tunica externa, pretty much the same on both sides. Endothelium of the internal tunica, pretty much the same. And then the connective tissue that's going to cover it. The big difference is this tunica media. That muscle lining in the middle is very thick in an artery and it's thinner in a vein. Also, you'll notice that valves are only present in veins. Valves are not present in arteries. And then lumen is labeled.
Mrs. Moody?
Yes?
Just a reminder to the high school students, if they aren't a player and they're going to the pep rally, we need parental permission.
Okay, thank you.
Thank you.
So the lumen is the opening. The lumen is the opening inside of the blood vessel, whether it's an artery or a vein. Basically, this just shows how there are smooth muscles that are around the arterioles. They're going to help to push blood through. Capillaries are your smallest vessels. They consist of a layer, very thin, a layer of endothelium. Substances are exchanged at the capillary level. So their lining can't be thick. If it was thick, stuff wouldn't go through it. So capillaries, one thin layer so that materials can exchange and go through. So that capillary permeability varies from one tissue to the next depending on the cells that it's next to. For example, liver, intestines, organs that don't have as much muscle are going to generally have more permeability, more exchange than some of your other areas or other organs. So there's a picture of your capillary. One layer, that's it. Nothing else to it. You can see how fluid, tissue fluid, can go in and out. There's little slits for substances to leave the capillaries. And then here's an actual picture, microscopic picture. I can see blue vein, red artery, and then my capillaries are going to be in between where they meet up at. Capillary density also varies greatly. Certain areas are more dense with capillaries. They have a lot more than other areas. Metabolic activity, muscles, large muscles need lots of blood flow. So your leg muscles, being large and used a great deal, are going to have a high density of capillaries. Your lungs are also going to have a high density of capillaries. Pre-capillary, sphincter muscles are those circular muscles, can regulate the amount of blood entering the capillary, and it's controlled by how much oxygen is in the area, if blood is needed there or if it's needed somewhere else. The capillary beds in less important areas, if you're not needing oxygen or something at a specific time, basically those capillaries, those pre-capillary sphincters can kind of shut off an area if it's not needed at that point in time. So, blood comes into the capillaries. They have high concentrations of oxygen. It's picked up oxygen, has nutrients, some electrolytes, and those are going to diffuse out of the capillary wall and into the tissue and cells that surround it. Plasma proteins are going to not pass through capillaries. They are going to stay in the blood because those proteins are too large, they're too big to pass through your capillaries. So, things that affect exchange in the capillaries. There's different kinds of pressure. Hydrostatic pressure drives the passage of fluid and very small molecules out of the capillary by diffusion, basically. So hydrostatic, fluids, small molecules, diffusion. At the vein, and this is at the arterial end. At the venial end, osmosis, which deals with water, due to osmotic pressure, the water that's in the blood, causes much of the fluid to return to the bloodstream. So hydro deals with water, osmosis deals with water. The difference is hydrostatic is typically at the arterial end, and osmotic pressure is going to be at the vein end. lymphatic vessels are going to collect excess tissue fluid. They can dissolve it or return it if it's needed. So if I look at the diagram of kind of all of this put together, I have an outward force of 35 millimeters of mercury. I have an inward force of 24. So I am going to have hydrostatic pressure, osmotic pressure. I'm going to have both. But what happens, the movement of material is decided by the differences in those. So here I can see my outward arrow is bigger than my inward. I have a net outward pressure, so fluid is going to go out. This is at my arterial end. When I get to my vein end, this is when I'm picking up wastes and carbon dioxide, things like that. Now my inward force is bigger than my outward force, so now my capillary is going to pick up stuff from the outside. So my arterial end, I want to shoot out the oxygen, shoot out the materials, the nutrients to the tissues that surround it. At my vein end, I want to pick up carbon dioxide, waste, things that need to be carried out. And that is how that exchange actually happens. And I am going to stop there before I start the next section.